Welcome everyone to this, our second PSP Association Research Information Day. We know from your feedback to the association and from our day-to-day experience as clinicians that research into the causes, manifestations, care and treatment of PSP and CBD is a very high priority for people living with these devastating conditions. The responsibility of the research committee is to promote on your behalf the very best research across all elements of laboratory, clinical, and health services research. In the UK, we're lucky to have a cadre of outstanding scientists and clinicians and a growing community of early career researchers dedicated to finding answers to the key challenges faced by people living with PSP and CBD. Today, our faculty will provide a broad update and overview of much of the research currently funded by the PSP Association. All this research depends on your determination to make a difference by increasing awareness, by fundraising, and by promoting understanding of these disorders. Your efforts have made today's agenda what it is. In addition, we must address the challenges of translating these advances into improvements in the day-to-day experience of people living with PSP and CBD. The final session will focus on some of these issues and particularly on your involvement with our research. Your voices inform how we communicate our research to change life for the better for everyone living with PSP and CBD. Thank you all for joining us, and thank you to all the speakers and session chairs for your contributions today.
And support from PSP Association members and families over the years. So can you hear me? Thumbs up. Yeah. Okay. So I'm now going to try and show my slides. So you can see my slides now? Yeah, okay, great. Good. Whoops. Okay, so I'm going to give you a little, it's a whistle-stop tour through what we've done. So a little bit of background, talk about what the study is, progress, what we've discovered so far, how this is going to help in developing new treatments, which is all obviously the aim of all of the research we're doing, and what the next steps might be. So the background to this really is sort of a clinical observation, our clinical experience, that PSP is quite a variable condition. So sometimes when we see people in clinic, people have quite mild problems that change quite slowly over time. Sometimes people develop more problems with a moderate pace, and sometimes people develop a lot of problems quite quickly. So that's represented here by the green, orange, and red lines representing time and severity. And of course, what we're really interested in is why there is this difference between patients with PSP. So can we understand the mechanisms behind that? And can we convert someone who has a rapidly progressive disease course into someone who's got a slowly progressive disease course? Can we predict what's going to happen at the baseline? And how can we measure and track what happens to patients over time? So with the support of the PSP Association, starting in 2014, we set up a longitudinal study that was initially tracking PSP and cortico-basal syndrome, which we called the Prospect study. I mean, this wasn't original, so similar studies had been carried out in Parkinson's disease, were being set up at that time, and similar studies in motor neuron disease, ALS, and Alzheimer's disease. So a lot of conditions, a lot of brain conditions were mounting similar studies because of their importance in taking forward research. As time went on, we sort of added extra things to it. So we added a cross-sectional study. So this was with the encouragement and support of the PSP Association. So rather than just patients coming up to study sites and being assessed, it enabled us to make contact with people remotely who either lived in remote locations or may not be able to physically come up to study sites, who are able to complete questionnaires and send in blood samples. MSA was added in with the support of the MSA Trust, led by Professor Henry Holden. MRI scans were added in a sub-study led by Professor James Rowe. We added in more CSF and biomarker measurements that we were carrying out at UCL. And then we were funded to continue the primary study, which we called the natural history study, and then continued in-depth assessment of new patients, the longitudinal study. And this is sort of an outline of the sort of arms of the study. So obviously everybody who enters the study completes, reviews the information and agrees, consents to be involved. In the longitudinal arm, people come up for study visits where they have scans, blood samples, questionnaires, examinations. And in fact, I always kind of think of this like a clinical trial, except without a drug treatment. And the cross-sectional arm where a similar approach is taken, but remotely. And again, through, as Alistair alluded to, through COVID, we ended up doing a lot more things remotely than we had done at the outset of the study. So we started off with 7 study sites in England and Wales. So the initial sort of PIs involved with it: Nigel Leigh, Michelle Hugh, James Rowe, Alistair Church, Alex Gerhard in Manchester, now kind of replaced by Chris Kobelecki, and David Byrne in Newcastle, now replaced by Nicola Pavasian. To be honest, there has been so much enthusiasm and support from this study from patients and carers and families and study sites. I've been kind of amazed by how much support there has been for this and how this has expanded. So this has grown from 7 to 29 study sites in England and Wales. And actually, as of yesterday, we have 1,329 participants in the study. And the map on the right shows the regions of the UK that contain a study centre. But actually, lots of these study centres are seeing people or recruiting people from a wider geographic area, which means that quite a lot of England and Wales now are covered in the study. And there have been separate PSP initiatives in Scotland that have been running. So this is a list of the study sites, which I'm not going to read through, but are available on the slides, which I think will be distributed. So these are currently the 29 study sites in the study. Just recognize our coordinating team based at UCL. So currently our coordinator is Rihanna Fumi, with Marta and Megan being involved in organising patient assessments, and our clinical fellow currently working on this is Dr. David Vaughan. So in terms of progress we've made with this, we have 810 PSP, cortical syndrome, and atypical Parkinsonism patients, in addition to MSA patients and controls. The first study, the natural history study, that has closed, and we published a baseline paper in 2019, and a follow-up paper is close to publication, being led by James Rowe on the follow-up of initial cohorts. The longitudinal cohort, the second in-depth assessment cohort, we have a target of 180 people, and we're currently up to 137 patients who've been recruited in that. The cross-sectional study, light touch, the target was 500 participants, and we're now up to 533 participants. So that's, you know, a lot of work from a lot of people, and despite the sort of setbacks of COVID. In terms of biosamples, these are listed here, and all of these, you know, biosamples that people have generated or donated are extremely helpful for taking forward research, and this includes cerebrospinal fluid collected through lumbar puncture, DNA samples, blood samples to look at proteins in the blood, plasma and serum, MRI scans, and part of the study is that we ask people if they're happy to register as brain bank donors. That's obviously quite a big step to date, but a huge amount has been learned about MSA, cortico-basal syndrome, and PSP through post-mortem donation. And unfortunately, people in the study do pass away, and currently 85 people have become brain donors who've been involved in the study. So briefly, what have we discovered? So obviously, there's not space to go through this in detail, and some of this has been covered in previous research days and research updates. But the diagnostic criteria for PSP were revised after we started the study, which kind of expanded the definition of PSP. So in addition to classical PSP, some patients with PSP present initially with a very similar presentation to Parkinson's disease and subsequently develop PSP features. And some patients present with a primary problem with walking, where the feet get stuck, they get freezing of gait as they walk. So these are 2 different types of PSP. And actually, when we added that into the study, into the categorization of cases, we actually doubled the numbers of PSP patients that were identified in the study, suggesting that PSP may be twice as common as was previously thought. We worked on a biomarker that's been widely used across a number of different diseases called NFL, neurofilament light. And this is a baseline blood test that can predict future progression. So that's obviously very useful for trials and understanding progression. We've discovered a genetic variant in a waste protein system within nerve cells that may be important in determining what type of PSP people develop. We've identified a variant in a gene called LRRK2, which is previously known as a Parkinson's disease gene, which seems to affect the rate of progression. And this is of great interest because there are a lot of drug trials that are directed at this LRRK2 gene, suggesting this might be relevant to patients with PSP. More recently, there's been modeling of progression through looking at sequential MRI scans, and also now matching the accuracy of diagnosis in life with what happens if people have an autopsy examination. So this is the sequential brain imaging studies. So this is looking at thinning down of nerve cells, atrophy of brain regions in patients with PSP, and lining up different brain regions in the order in which they might be affected. And you can see this represented on the slide, on the diagrams on the right, suggesting that PSP starts in the lower part of the brain, then involves the deep bits of the brain involved with movement, the basal ganglia, and later on involves the outer parts of the brain, the cerebral cortex. So we can see there are different, as people get worse in clinic, as we see them year on year, there are different brain areas that are involved. And obviously modeling things in this way is very helpful for understanding how we could intervene and prevent the pathology from PSP from getting worse over time. This is looking at the accuracy of diagnosis in people who've been studied in the brain bank. So the left-hand diagram shows that there were 49 people in this bit of the study diagnosed with PSP, and when they were studied in the brain bank, 42 of them had PSP pathology, and some patients who we thought had PSP in life turned out not to have PSP at post-mortem. So that included patients with cortico-basal degeneration, CBD, which is very similar to PSP, but also one person with Alzheimer's disease and one person with MSA. And this sort of underlies the need to have better biomarkers to help with the diagnosis, so blood tests or brain scans to help with the diagnosis. And as we can continue analyzing data, we'll be adding in these sorts of markers with the help of James Rowe, looking at MRI, our colleagues at Queen Square, and people looking at blood and CSF biomarkers to try and find markers that help us to much more accurately diagnose patients with PSP. One of the things that we've done is make the data and samples available to other investigators. So essentially, external investigators
Hugh, I hate to stop you in mid-flow, but we are time up. So are you able to sum up for us? Thanks.
Okay. So I will just talk about the future of this, so that we're going to continue recruiting new patients until July 23, with a follow-up through to July 24. The main study will then close, but we'll continue some remote follow-up. One of the big things that's come out of the study is the delay to diagnosis in PSP. So there's an interval of about 3 years before people get diagnosed with PSP. So we're trying to develop a next study which really tries to aim to identify PSP patients much earlier in the disease course, which relies on sort of intercepting, if you like, GP referrals and early hospital assessments. I think that's going to be very important for future development of the disease. And we hope we'll develop future trials. We'd like to study PSP, particularly in non-European ancestry populations, and develop more biomarkers. So that's briefly the prospect study. And I'm really grateful to all of the patients and carers who've helped us, the funding support, coordinating team, all of our study teams and PIs around the UK, and look forward to completing this stage of the study and obviously maximising what we've done to try and help patients with PSP in the future.
Thank you very much, Hugh. That's fantastic. Don't forget, we've got question answers at the end of these 3 talks, so please put them in the Q&A box and we'll go through as many as we can. That's fascinating. Thanks, Hugh. Okay, we need to move on now. So our next talker is Professor Antonides. She's going to talk to us about measuring movement, I presume, in PSP, which I think is going to be very relevant given that sort of wearable technology is really coming to the fore now. So that's going to be great. Are you there, Professor Antonides?
Yes, hi. Morning, Alistair. Very nice
Wonderful.
to
Good morning.
see you. Let me just, this is not letting me share it properly. Just bear with me. Okay. All right. Can you see this okay?
That's great. Yeah, that's good.
Okay.
We can hear
Wonderful.
it.
Thank you. Right. Good morning, everybody. It's a great pleasure to be here. It's fantastic to see so many people joining today. And I'm looking forward to hearing from all the other colleagues as well. So what I'm hoping to do in the next 10 minutes is tell you a little bit about a study we've been running here in Oxford, which we've been trying to quantify progressive supranuclear palsy. And give you a little bit of an insight for those of you that are not part of the study, what it involves, and give you, if you like, within a few minutes, a very general gist of some of the recent results we've been finding. So the study I'm running here in Oxford is called OxQUip study, and in short for Oxford Study of Quantification in Parkinsonism, as you can see here. This is essentially a longitudinal study where we're trying to follow up people both with Parkinson's and PSP very, very closely. So every 3 months for about 3 years. So that's a lot of visits for my team here that have been doing a great job. And the aim of OxQUip has been to be able to precisely measure these subtle abnormalities that we know exist in the timing, speed, and coordination of a range of movements. And I'm very grateful to UCB that has been providing us with funding, both for phase 1, which has been the in-clinic face-to-face, but also for the remote telemedicine we've been doing in the last year and a half. And what I want to show you here is a little bit of the type of test that we've been using as part of OxQUip. So we look at, for instance, your eye movements. We have been using different oculometers, so different ways of measuring how you move your eyes. We've been measuring how your finger tapping works, so how you tap from side to side, and we've been using reaction time as one of those parameters, if you like, that have been tremendously helpful and interesting and promising. We've also been using a different number of sensors that we place on your body, and I'll show you in the next few slides how those have been providing tremendously helpful and insightful information about the disorder itself and how each one of you could be tracked over time. But also we've been very keen in trying to understand how we can measure your cognitive abilities, for instance, your memory or your ability to perform a task, from sort of making it from pen to paper into what we call the cognitive tablet. And I don't know if you can see my little video here, is where one of our participants is just asked to connect the dots here, going from the largest square to the smallest square, or from the larger circle to the smaller circle, and so on. For those of you that haven't had, for those of you that have not been in our study,
I thought it started after,
and haven't sort of experienced sort of eye movement recording, this is what it looks like when you come into clinic. We just place a simple oculometer on you, so this device that sits on your forehead. And you can see that is very easily measured by sort of 10-15 minutes timing. It's not invasive and is very easy to use. For instance, here is a very simple task where we ask you to just follow a dot from side to side, and we can see how quickly or how slowly you can move your eyes. And of course, people always ask, what does this data look like? Well, it's a very complicated set of data. For instance, here you can see, don't worry about the details of this, it's just to show you that we can track very, very carefully how you're moving if we ask you to do what we call a 2-minute talk. So let me just show you 3 important areas and 3 important findings, rather, that we've been sort of looking through from the data that we've collected in the last 3-4 years. So this is one study that we published this past year in BMJ Neurology Open, where we've looked at all the different parameters, if you like, from the clinical rating scale, so these questionnaires that all the neurologists that you see ask you when you come and see us in clinic. And what the take-home message for this is, and again, don't worry about all the graphs and the complicated numbers here, is that if you look at this little graph here that I'm pointing, called PSPRS gait, it's essentially one of the main areas that we found out that was of great interest. And what this says is that with these clinical scales are very helpful. And in fact, we have found that within 6 months, we can pick up a significant change in someone's gait. So how a person is walking, just by asking you a number of questions when you come to see us either in clinic or in lab. And that has been tremendously interesting, especially in trying to understand how we can use these questionnaires that you've been asked every time you go to see someone and are followed up. So the second thing I wanted to show you and share with you is something we found that we call the strategy adherence. And essentially what we found here is very much based on the cognitive tablet that we've been using. And what I mean by a cognitive tablet is by basically doing a task that is on a tablet as opposed to asking you to use a pen and connect something on a paper. And in brief, this is the so-called cancellation task. So this is a test that is used commonly in psychology to assess patients, and it's traditionally been carried out by pen and paper. And what we did, we moved this pen and paper test to a computerized version, and we asked our patients to follow whatever way they wanted to choose, if you see here on my screen, which ones the fruits were. And within these fruits, we had vegetables as well. So you're presented with fruits and vegetables. And we asked them to choose which ones they thought they were their fruits. And what we found out is that in people who didn't have a diagnosed neurological disorder, and they didn't have Parkinson's or PSP, we found that they could solve out systematically and choose in a very, very structured way, if you like, where the fruits were. But what we did discover is that a large fraction of people who did have PSP did not follow a strategy from the outset. And the proportion of those individuals with PSP who sort of lost the strategy of deciding whether I go from left to right, from top to bottom, and decide where the fruits were in this particular screen rapidly declined. And we find a significant change even after 3 months of following individuals who had PSP. And essentially, just to make it really simple, we just looked at the patterns. So what you see here, I hope you can see that these different patterns are very different from each other. Some of them are very structured. So some of you chose to look at the fruits line by line. Some of you looked at them from top to bottom, and some of you had sort of different ways of doing this, but some of you have very random ways of doing this. And it's been very interesting that looking at this and analyzing this very, very carefully, we realized that the number of PSP participants that were losing sort of the structure and how they were following this particular way of choosing the fruits does change as the disease progresses. And I guess that's a very promising way and a very easy test to use in clinic, and especially in clinical trials. And the last result I want to share with you is having to do with the different sensors that we've been using in placing all over the patient's body. So the first bit we found was that if we use different sensors, for instance, I don't know if you can see my screen because I can't see you, when using sensors such as the one I'm wearing here, so watches that are worn on both hands, a sensor that is placed at the front of your chest, one at your lower back and one on each foot. Therefore, we can have a very good idea of your movement, how your body is going, and how your body is sort of responding to different things that we're testing it. We found that by asking someone to do something that we call a 2-minute walk. So we ask you to walk a stretch of in a dedicated corridor here in the John Radcliffe in Oxford for 2 minutes, and then turn around and come back as many times as you like within those 2 minutes. We discovered that we could discriminate patients who had PSP and those patients that had Parkinson's or something else. And the way that we did this is by asking people to walk for 2 minutes, asking people to get up from the chair and walk to the end of the clinic and come back. As you can see, one of my DPhil students here demonstrating, walking to the end of the clinic and coming back. And then by starting for just 30 seconds with your eyes closed, just to see if you're swaying. And that's swaying both forwards and backwards or right and left. And as you can see, Lily here is wearing all the sensors. I don't know if you can see that very well, but she's wearing 2 watches, 1 on each arm. She's also wearing this sensor at the front, at her chest, at her lower back, like a belt, and 1 on each on her feet here. And that has been really tremendously helpful in carefully helping us to accurately and sensitively figure out which patients had different ways and different patterns of walking. And of course, this led us to the final thing, my last couple of slides here, about how we can use these different sensors and this fantastic technology that has been sort of developed over the last few years to be able to track how each one of you progress, especially by testing you when you walk and how you stand, and what type of sway you have. And I've just shown you the different types of movements that we ask you to do: the walking, the getting up from the chair and turning around, but also standing with your eyes shut and with your eyes open. And what we attempted to do for this last study and these latest results that I'm presenting you here is to basically track the progression of walking and swaying and figure out which sensors, if you like, would be helpful in doing that. And what we did, again, don't worry about all the little details here, it's just to show you that the pipeline, so the way that we go to build a model, a computational model, as we call it, can be quite tricky if we don't decide from the beginning how we're going to attack the question we're trying to find out, but also the type of sensors that are being used. You probably know there's a lot of technology out there. There are different companies advertising different technology. And I think one of the things that we need to be careful from a researcher, clinician researcher point of view is that we really need to be careful how these sensors are used and what are they actually giving us in terms of the data. And what we found here is that
I'm very sorry to interject, but I'll need to run to time. So
I'm finishing in 1 minute. Yes.
Wonderful, thanks.
What we found here is that using that methodology, using technology, for instance, all over your body, like I've just shown you, and using clever mathematical models. Don't worry about the details again. It's just to give you a glimpse of the amount of data and the amount of collaborative work, if you like, we need to be doing to be able to be arriving at this stage. We've been finding some very exciting and interesting results. And one of it is that by measuring the gait and speed of PSP patients using these movement sensors, we found a signal that predicted deterioration before standard clinical rating scales. And I think this is quite important finding for diagnosis and further research. And it's been tremendous having the support from all of you, from PSPA and all our international collaborators as well. I'll leave it to that. Thank you very much. I'd like to thank our fantastic patients and their families for letting us into their homes now, with following you through home monitoring, and of course, the tremendous support from PSPA, who have been supporting us every step of the way. Happy to
thank you very much. Thank you very much, Kristalina. And don't forget questions in the question answer. We will answer those afterwards. That's great. Lots of opportunities for the future, I think. Okay, so we move on to Dr. Stockton. Are you there, Dr. Stockton?
Yes, hello. Let me just.
Wonderful. Hi, good morning. Thanks very much.
Great.
Okay, I'll hand over to you. Thanks.
Hi, so I'm Catherine Stockton. I'm one of the clinical fellows with the James Rowe and Tim Rittman in Cambridge. And what we want to tell you about is a study called the PiPPIN study. Next slide, please. So what we're looking at is a family of the related conditions, which is predominantly PSP and its cousin, the cortico-basal syndrome, and another one of the cousins called frontotemporal dementia. There's a lot of overlap in how these diseases present. As you've heard from Hugh, that there's different ways that we can see how they come across to us. And what we want to capture is how many people within an area have these diseases. So what is an epidemiological study? Essentially, it's a head count of everyone with PSP, cortico-basal syndrome, and frontotemporal dementia in the area of Cambridge from Norfolk. And why do we look at it this way? What we want to do is to ensure the results are relevant to everybody in this. So it's extrapolated that we can say that these people in this area have these diseases. We know what the population is in this area, and we can then say, well, if there's this many people with this disease in this area, how many people across the country might have the disease? So what we want to capture, first of all, is how many people get the disease, at what age, how many men, how many women, and what symptoms that you have. So when we talk to you, what symptoms do you have when you come to see us? What signs do you have when we examine you? What is the impact of the disease on yourselves and on your carers? And also things such as, what is the delay to diagnosis? How long does it take us to see you in the clinic? Or as we've seen from Hugh, there's a delay. We want to know why it is, and what is the overall outlook for the disease. So next slide, please. Next slide, yeah. So on the left, I've got the conditions that we look at in the study, and the PiPPIN study has been running since 2013. It's been going in 3 phases, each at 2 years, and what we have recruited around 200 patients at each time. So we found that there's about 200 patients in the first phase back in 13 and 14, another 200 in 2017-18, and in this last phase, which I've been recruiting in, it's another 200 patients roughly. So what we do is we look at it in 2 tiers. The first thing we want to really capture is how many people have the disease in the area. So as you can see on the right, we're getting quite a good range of people across the whole of the area. It's not just focused around the cities. We want to complete some blood tests alongside that, which is just to make sure that we can do some further tests on that, looking at the genetics and other inflammatory markers that we can see. The next thing we want to do is to get a bit more of a detailed idea as to what is going on with the diseases. We want to look at the imaging, so brain scans and behavior. So we're doing some cognitive tests, similar to the ones that you get when you come to clinic, maybe some computer tests as well. And if you happen to be in Cambridgeshire and Norfolk and haven't yet heard from me, then do please get in touch, because I'd love to make sure that we actually get a representative data set that includes everybody who has the disease in the area. Next slide. So why is it important that we capture the whole spectrum of data? Well, everyone is different. And what we see is that we can look at the individual diseases, like in the bottom left, you can look at PSP in blue or cortico-basal syndrome in green or frontotemporal in red. But actually, when we look at them together, we see there's a lot of overlap in how people present. And so we group them together so as to not miss the people who present differently. Next slide. So looking at the individual diseases, just briefly, don't focus too much on the details, other than we know that PSP is a movement disorder problem predominantly, and the movement disorders are listed in blue. So we can see that it's a problem mostly in the midbrain, and it starts with problems with rigidity, movement, and eye movement difficulties. But we also know that patients do have difficulty with their thinking, with their behavior and personality, and apathy, for example. Cortico-basal syndrome, similarly, is a disease of the back part of the brain predominantly, but it has the movement disorder problems as well as the cognitive problems. Whereas frontotemporal dementia, which can present in a similar way with cognitive problems, similar types of cognitive problems, can also have movement disorders. And what's significant here is that we know that some of the patients with PSP can present looking like a cortico-basal syndrome or a frontotemporal dementia patient at the start. But only as the disease evolves does it become apparent that actually it's the PSP that's driving the diagnosis. And we don't want to miss these people. Next slide. So what we see on the right is the diagram which shows how much overlap there is in each of these diseases. So you can meet the diagnosis for PSP, which is the blue corner, but you can overlap with the diagnosis for frontotemporal dementia or cortico-basal syndrome. Next slide. One of the things of looking at the diseases as a global perspective like this is missing and counting all the symptoms that we get when we see you in clinic, rather than focusing necessarily on the diagnosis that we see, is that it can show things that come out which aren't necessarily part of the initial diagnosis. So one of the things that Claire Lansdowne showed in our group earlier in 2019 was that actually apathy is one of those symptoms that can have a significant effect. We see apathy as the get up and go, whether or not you are still driven to get up and do things on a day-to-day, or whether you prefer just to sit and watch and do the same thing on a go-by-go. So it's common in PSP and cortico-basal syndrome. We know that it happens early on in the disease, and we've also seen that actually it affects your outcome. It can affect how you are from day to day, and we know that people who present with more severe apathy at the beginning have a worse outcome overall. So one of the things that we're looking at as a group is to see whether or not, if we treat apathy, whether or not we could influence survival and outcome, and also, importantly, whether or not it can influence the day-to-day, whether the burden of the disease can be improved by having a treatment for this. And this is a study which you'll hear about later on from James Rowe called NORAP. So I'll leave that to him to tell you more. Next slide. So where are we with PiPPIN so far? So we have, since 2013, recruited 545 patients. We've seen in the last 2 years, 145 new patients, plus nearly 60 who we've seen from the first 2 studies. So that's 8 from 2013 who are still with us, and a further 50 who are from the 2018 cohort. So we've got 100 controls as well. And what we like to make sure that we're looking at is also the ethnicity and deprivation data, which matches, to make sure we're getting a good representation for everybody in the area. So far, we have 200 PSP patients, nearly 100 cortico-basal syndrome patients, and 120 behavioural variant frontotemporal dementia patients, and the other ones listed are also part of the frontotemporal dementias groups. Next slide. So again, what we want to know is, are we capturing everybody in the area? Are we making sure that we're getting the groups together so we can make sure that we're not missing people, and that the data is not missing things that would have an impact on health inequalities? We have a population of around 1.8 million in Norfolk and Cambridgeshire, and we know that the average ethnicity for the area is 92.7%. But in the older population, actually, it's closer to 96%. So we are capturing the right ethnicity for the area, although it's very selective because it is a predominantly white British region. On the right, what we are looking at is whether or not we're getting everybody who matches the health inequalities that are there. So we're looking on the grey line is the number of people that we'd expect from each of the different deprivation groups. There's a list that says if you're in the dark blue area that you live in a low deprivation area, or if you're in the dark red area where you live in a high deprivation area. And what we see is that actually the people we're missing are possibly in the lowest deprivation, which leads to the health inequalities. And what we really want to figure out is what is an element that's affecting this, and how can we change that to improve that for everybody. So next slide. So what we want to know is, who are we missing? Is it just the people who are finding it difficult to get a diagnosis for whatever reason? Are they delays in coming into the clinic? And we also, who are we missing when it comes to those who die before they get a correct diagnosis? Next slide. So this is a little complicated on the right, but essentially PSP is the 4th line along of the pairs in the graph, the red and green, and cortico-basal syndrome is in the 5th slide. And you can see that from the time from diagnosis up to the time of admission to care home or to dying is significantly lower, is shorter for the people with PSP and cortico-basal syndrome compared to some of the other diseases. And as Hugh alluded to, there is a delay to diagnosis of around 3 years. And what we need to figure out is how can we capture the group more significantly earlier on. So one of the things we're looking at, so some of the things we're looking at within this study include the genetics, things which you'll hear about from Ed Jabari later, about how some of the genetic changes will be affecting the disease outcomes. We're looking for blood tests that can help with the diagnosis, so other biomarkers which can help with getting the diagnosis sooner, earlier.
Catherine, hi, I'm sorry to interrupt in mid-flow, but could you sum up in the next minute, please?
Yeah, that's in the next slide.
Thanks
okay.
so much. Thanks.
So what we also see is the impact of cognition is under-recognized. We want to really look at how things are impacting from a cognitive perspective, and who needs a care home, why do they need care home, and why do some people live longer? Skip 2 slides. So in summary, what we really want to know is that everyone is involved in research. That's really important to us. We want to make sure that everyone is engaged in the research, and so that it's actually relevant to all of the people in the area. We look at it as a transdiagnostic approach because we really don't want to miss people who have the overlapping symptoms or who have a misdiagnosis in the early stages. We want to aim for biomarker tests that can predict the outcome and to show us how things are going to look as the disease goes. If you have any information or referrals, then please do send information to James Rowe. Next slide. And if you live in the Cambridgeshire and Norfolk area, then please do get in touch with me, as I would love to include you in our study. So those are my details. And thank you to James for working with him, and all of the patients and carers who take part in the study, and all the sponsors as well. So thank you.
Thank you very much, Catherine, and thanks very much to all our speakers. Time just seems to fly. I'm sorry to cut you all short. I think we could have all listened to all of you for a lot longer. We have some questions. Obviously, if anybody wants to put any more in the chat box, please do. So I'm just going to go through them in the order that they came in, if that's all right. And if whoever feels most up to it, please reply. So this one is really, I think, open to anybody. Everybody's one from Joanne wants to know about the link, if at all, between Viagra and PSP. Apparently there are some studies that show that Viagra reduces tau burden. But there be any concerns about you recommending your patients take Viagra if indeed that link is true? Anybody want to pick that up?
Yep. So I'm not aware of any trials that are planned for this. So I think one of the problems with stuff that appears in the press is that a lot of this is based on sort of preclinical models, so work in mouse models or cell models, and that may not be immediately translated to like patients. And what works in a mouse may not work in humans. So there's kind of quite a lot of work to do to work out if it's worthwhile to use. I mean, in terms of using Viagra for erectile dysfunction, actually that's quite a common thing in MSA patients. So MSA patients often do use Viagra, and the main sort of side effect is low blood pressure, which is a particular problem in MSA patients, but many patients do seem to tolerate it quite well. So that's the kind of thing to watch out for. But it is possible, particularly actually GPs, to prescribe Viagra for people with erectile problems, but we wouldn't recommend it for modifying the course of PSP at the moment, as far as we know.
Thank you. Yeah, indeed. Unfortunately, many trials that have shown promise in mouse models, when they've been projected into humans, have sadly not come up with a positive result. Okay, so thanks. So we have a question from Hamed. How can we take part in these studies? I mean, I think we've sort of answered that to some degree, but perhaps, Catherine, as I cut you short, perhaps you could just put another quick advert for if you live in Cambridgeshire or Norfolk, or whether you would recommend people move there if they want to take involved.
So we're looking very much so for patients who are living in Cambridgeshire and Norfolk who have cortico-basal syndrome and PSP. So we want to make sure everybody is included in this area. If you are part of the PiPPIN study, you can also be part of the Prospect study as well, because they are overlapping, and we try and incorporate everybody in that area. So we try and get all people to be as part of as many studies as they're willing to take part in. So do contact us, get in touch with us, as we'd love to have you with us.
Yes, I mean, I think everybody with a neurodegenerative condition should have the opportunity to take part in a trial. You just need to contact your local department, and if they're not associated with any trials, then I'm sure they will put you through to those that are closest to you, and obviously the PSP Society will be able to direct you appropriately. And Professor Antonides, are you still recruiting for your studies?
Yes, we're recruiting until next month. And in fact, I was going to say that the PSPA website, I find it is very useful in directing people and seeing which area of the country you're living in to sort of help direct people to, whatever is closer to them.
Wonderful, thanks. Yeah, I mean, I think even if they don't directly help you, I think that it's good to fight back and feel that you are doing something to advance our understanding of these conditions. That's the only way we can move forward. So please do step forward and take part in trials. So this is one from an anonymous participant. It's probably one for you, Hugh. Can NFL blood test be used as a diagnostic marker for PSP patients and or to aid the diagnosis in patients who have suspected or possible PSP?
Yeah, well, this is not in use at the moment. I think that, when we're looking at sort of diagnosis or progression, we can see kind of differences between the groups, but often there's an overlap in the values between patients and people who are unaffected by the condition. And I think that there is a lot of interest in kind of implementing this as an NHS test to help with early diagnosis of these conditions. In a way, that's kind of what we're exploring with our research. I think that I think that the future of this actually is probably going to be integrating several sources of information. So maybe integrating a biomarker with some clinical data with a scan, and to try and get an algorithm and digital data as well. And I think that, you know, getting an algorithm which kind of improves the diagnostic accuracy. So the other thing with NFL, although it looks like a very good biomarker, it does increase with age. So actually it goes up in the normal unaffected population every sort of decade, which makes it quite difficult to interpret in people in their 70s who present with neurological conditions. But I think that this will come into use as a marker. I think it will probably be used in clinical trials, so they'll probably use it to help understand the patients who are going into drug trials, and then it may well come into use as a clinical marker, but not at the moment.
Okay, thanks very much. Yeah, but I can see a world perhaps somewhere in the future where you can monitor your own levels every year, and when they start going above the sort of your age match control, maybe you might want to start seeking help and perhaps start doing some online tests of your eye movements and hand-eye coordination as per Professor Antonides' trials, and perhaps even diagnose yourself at home, and maybe your watch will tell you that you're not swinging your arms as much, etc. And you could see how you could put that all together. Okay, so next question. Is there a stage in PSP when it's too late to join the study? I think the quick answer to that is no, I don't think it is it? If you're up for it, then getting in contact with your local centre. Okay. Last question. Is there no way to perform a procedure on the brain to reduce the amount of tau? Even if that reduction is at all impossible, can a small amount be removed or try and prolong the quality of life? So I don't know who would want to pick that one up.
Yeah, well, one of there were trials that ran that completed about 18 months, 2 years ago. We're using antibodies to try to reduce the amount of tau that's in the brain and sort of shuttling between nerve cells. So those trials were negative in that they didn't make a clinical benefit. There's a further trial that's open at the moment which switches off the message from the gene that makes tau, which Boyd Ghosh is recruiting to, I think, still in Southampton. I hope he's on the call. So that is very directly directed towards reducing the amount of tau in the brain. I mean, it's a very early stage trial, and there are very small numbers of patients who are being recruited to that, but it is something sort of actively being looked at. But it's kind of, if you like, quite kind of cutting edge in terms of the sort of experimental therapeutics approach. And in fact, the tau lowering agent that's running in Southampton, it's a Novartis study. Actually, the drug is administered by lumbar puncture, so patients who are involved with that have lumbar punctures to do that. So it's obviously quite a demanding thing to do, but as you suggest in your question, we think this is a good approach. We think this would potentially be a good approach to try to improve quality of life and the sort of way people are affected by PSP.
Thank you very much. Thanks. So that's the end of our questions. I just want to thank our three speakers for the first session. It's been a great start, and I'm sure the day will continue in that vein. So thanks very much, everybody. We've got to